Okay, great. And so the third speaker in this session will be Dr. Jordan Ubbens. And as I mentioned, he couldn't be here, but he quickly made a video just right before the session so that he could join us virtually. So Jordan is a research associate at the Global Institute for Food Security. He completed his bachelor's and master's at the University of Regina, and then we recruited him here really as, in my group, one of the first PhD students in the PERC program. So Jordan's really been with us since the very beginning and has made really fundamental contributions and has been, a really excellent team member. Jordan's main interests is in machine learning applications in plant genomics and phenomics, and a particular focus on the identification of stress-tolerant varieties. And so we will hear from Jordan now.
Hi, everyone. My name is Jordan. I was supposed to be there in person today, but I woke up with a case of the sniffles, so I hope that this online Zoom format makes you nostalgic for a time when all of our conferences look like this. So I'd like to talk about genomic prediction and genomic selection. So a little bit of background first. Genomic prediction is the practice of predicting an organism's phenotype using a collection of whole genome-wide markers, usually SNP markers. And then genomic selection is the implementation of that, where in a breeding program you want to select breeding crosses based not on what their phenotype is, but what the estimated breeding value is based on these techniques. We usually do this kind of thing with linear models, such as GPLUP, or Genomic Best Linear Unbiased Predictor, or other sort of related linear models. In practice, they seem to work quite well, despite their sort of lack of expressive ability as a model. Back in the early part of the 2010s, there was a lot of sort of anticipation that powerful nonlinear methods like neural networks were going to increase the rate of gain for genomic selection, because these nonlinear models, being much more powerful in their abilities, are actually able to predict nonlinear effects, such as epistasis, which is a nonlinear effect between two or more markers. So there's a lot of excitement about this, but then as the literature sort of matured and we accumulated evidence, it looked like these models didn't actually offer any advantages at all over the classic linear models like genomic BLUP. And the reaction to this was sort of, oh, well, but I got really interested in why this is, because there's sort of two main implications. Either A, epistasis doesn't exist for most of these agronomic traits, i.e. they are actually strongly linear additive in nature, or B, nonlinear models like neural networks decide that they don't have to estimate nonlinear effects, despite the fact that they could, so that they decline to. And so we wrote a paper on this topic, which was the sort of the first exploration of this question. And we proposed in our hypothesis that the answer is actually the latter. So despite the ability of these nonlinear methods to predict epistasis, they decide that they basically don't have to. So they're able to use sort of large broad stroke effects like ancestry, patterns of linkage, to sort of develop a concept of the phenotype without having to resort to actually estimating any marker effects at all, epistatic or otherwise. So what we did in the paper is we trained a neural network while blinding it to the alleles themselves. So it was only able to access which locations in the marker data to individuals were the same, without actually ever knowing what allele is there. And we saw that when we blinded it to the alleles in this way, it didn't actually matter. So the predictive performance wasn't affected at all. It wasn't deleterious. So that's a really interesting result. And so we proposed this as a explanation for this effect. And so that was in 2021. Recently, I've been really interested in the question of, so if it's only the broad stroke effects like ancestry that really matter, then we could probably compress these data sets to very, very small bits of information and still retain a lot of the predictive performance of linear models or nonlinear models equivalently, i.e., there's not actually a lot of information that's relevant for prediction. These genotype data sets are usually made up of tens or hundreds of thousands of individual SNPs. So here's some examples of some breeding and wild populations, as well as the number of SNPs included in them. The next column over is the number of principal components that explain about 90% of the variance. So this gives you an idea of basically assuming that the data lie on a linear manifold embedded in the space, how many dimensions would you need to construct about 90% of it? And then the last column is the most interesting one. So here we're using a existing intrinsic dimensionality estimation technique from the literature to estimate how many continuous factors of variance are there, assuming that these data cluster on a nonlinear manifold, a nonlinear subspace within that higher dimensional ambient space. And you can see that even though we have tens or hundreds of thousands of SNPs, the intrinsic dimensionality is usually only a couple dozen. So this lets us know that, like with other kinds of data, like image data, these data are actually quite small in the number of factors of variation. And it got me thinking, can we go even smaller? So all of these methods assume that the latent structure of this data is actually continuous. So principal components are continuous. The maximum likelihood is estimating continuous axes of variance. What about if we constrain it all the way down to just a discrete latent structure? So we can actually use something like this to compress it down to just a few bits of information. Here's a kind of machine that you might use for doing something like this. So this is something like a vector quantized variational autoencoder with a multi-head encoding and autoregressive or recurrent decoder. So this is a kind of complicated machine, but the idea behind it is quite simple. Basically, it's taking that huge high dimensional original data set and trying to compress it down to just a few code words. And the amount of code words will tell you the bit depth representation that we're compressing to. Here's an example of rate distortion for a few different data sets. So on the x-axis here, you see that we have bits. So this is the bit depth of that representation I was talking about. And then on the y-axis, we have something called distortion. So basically, this is telling you how compressible these different data sets are. The higher the line, the less compressible the data set is. And so you see on the x-axis, as we vary from 16 to 128 bits of information, we see that strawberry is actually very hard to compress. Wheat, a little bit easier, and then lentil and brassica carnata are on the bottom of the chart. Interestingly, our levels of compressibility here, they don't actually correspond to the extrinsic dimensionality of the data, which is the number of SNPs. Rather, they correlate to more interesting factors. So for example, all of these relate quite well to ploidy, and the amount of recombination also contributes to the genomic complexity, so that could be a factor, as well as you also see artifacts of the complexity or the diversity of hybrid ancestry in the levels of compressibility here. The strawberry dataset, for example, has a very wide range of hybrid ancestors in that dataset, versus the lentil is a diversity panel, and the brassica is a NAM population. So this is an interesting representation of basically relating how compressible the data is to both the genomic complexity as well as the diversity of the population. And so back to the main event here, I was very interested in, are we actually able to reproduce the performance of something like GBLUP or other models using these very, very small discrete latent representations. So here's an example from the lentil diversity panel where you see that using only 22 bits of information, we're actually able to be competitive with GBLUP for this particular trait. Taking a look at some different data sets, we have lentil wheat and Brassica carinata, and then we have corresponding to those the performance of GBLUP in Pearson correlation represented as the light-colored line. And then again, we have the bit depth of representation on the x-axis. So you see that with the lentil and wheat, we're matching the performance of using the full data set using only 128 bits of information. Another interesting case is the carinata population, which is the breeding population. You might say that, well, the lentil and the wheat is land races, the lentil is a diversity panel. So you might say that, well, by far the population structure explains most of the phenotypic variance in populations like these. So it's kind of not surprising that you're able to recover a great deal of the performance using a discrete latent representation, given that it's probably easy to compress population structure into this information. So carinata is an example of a breeding population without population structure, where all of the individuals are half sibs or full sibs. And you can see that with 120 bits of information, we're actually getting pretty close as well. So this representation isn't just encoding population structure. It can also include things like major haplotype blocks and other sort of artifacts like that. So a few takeaways. Firstly, we showed that genotype data actually has a very low intrinsic dimensionality. This is sort of intuitive, but we kind of articulated it quantitatively. And we showed that, at least with respect to the genomic prediction task, we can actually explain this with a discrete latent structure, which compresses it even further from something like a nonlinear continuous manifold. And we also saw that the compressibility of population is correlated to its genomic complexity, i.e. its ploidy, its recombination rate, or the diversity of hybrid ancestry, not to its extrinsic size, which again is that number of SNPs. So thank you for watching my video. I unfortunately am not able to take questions in person, but if you just email me, jordan.ubbens@gifs.ca, I would love to chat with you about this topic. Thanks.